Hello, today we will discuss about electroencephalography. Previously we have seen what is your electrocardiography, how the electrical activity of heart can be recorded and from which significant information about the physiological activity of heart can be taken away. So in this lecture we can see how this brain electrical activity can be captured, and it will help us. By capturing this, we can detect different types of disorders related to brain, nerve system, and also it will help us to understand the human behavior. Now we'll see our head or this human brain. It has a cerebrum, then we have cerebellum, and then medulla oblongata. Basically we are focusing on this cerebrum. This cerebrum that is divided into, it is divided into left and right hemispheres. So you can mark here. In this picture clearly it is visible that different lobes are there on this cerebrum. This cerebrum, it can be divided into different lobes, and the basically if you see from the top, we will have a left cerebrum and the right hemisphere of the cerebrum, left hemisphere of the cerebrum and right hemisphere of the cerebrum, and in between them there is a deep longitudinal fissure, and that two hemispheres, they are in contact for communication purpose, and that contact is through this corpus callosum, and each hemisphere further divided into this different lobes. Basically we will focus on these four prominent lobes. Those are frontal lobes, parietal lobes, then we have occipital lobes and temporal lobes. So depending upon these lobes, their functionality and they are also differs. So if you mark the frontal lobe, actually this lobe consisting of, or it controls the judgment, the problem solving, planning, behavior, personality, speech, writing skill, speaking capability, then concentration, self awareness and intelligence. So frontal lobe has the control of these different skills. Next is your parietal lobe. In this lobe, it controls perception and sensation. Occipital lobe, this lobe interprets vision, distance, depth, color and facial recognition. And finally the temporal lobe, this lobe controls language comprehension, hearing and memory. So now we have one idea that which lobe contribute to which skill of human being, or these lobes, they are due to these lobes are they are made up of white matters and gray matters. These electrical activities, we know that the gray matter and white matter, they contains numerous neurons, and these neurons, they have some your excitation and action potential has been generated. So these action potentials which has been generated in this gray matter and white matter, they are travel, or these changes of electrical activity we need to capture through this surface electrode which has been placed on the scalp. So you mark one thing, these surface electrodes which are placed on the skull, and the signal which has been generated here deep inside this gray matter, so there is one different layers are there. So this signal has to pass through this pia matter, then subcarotenoid space, arachnoid space, then dura matter, then skull, then we are placing our electrode on the scalp. So that is why the signal strength is reduced into some micro volt order. So to capture this signal, this electrical activity generated deep inside our brain at different lobes, as we have discussed in the previous slide, Hans Berger, a German psychiatrist, he has first developed this EEG, electroencephalogram, in 1924. The EEG, an electrophysiological technique for recording of electrical activity arising from the human brain. This helps us to diagnosing many neurological illness and diseases, epilepsy, then tumor, cerebrovascular lesions, ischemia, and problems associated with trauma. It also used in operating to facilitate anesthetics and to establish the integrity of the anesthetized patient's nervous system. A typical EEG display graph is a voltage versus time graph. So, we will see we have different time and voltage. So, the waveform will move, it is just like that, it is a voltage versus time graph, providing a real time display of ongoing cerebral activities. This electroencephalogram works on differential amplifier because we have placed multiple number of electrodes, that those electrode can be unipolar or bipolar, but we are considering the difference. If this is a bipolar electrodes, we are considering or we are amplifying those difference of these two electrodes or the signals captured through these two electrodes or different electrodes. So, recording the voltage difference between an active electrode and a reference electrode. This type of electrodes are unipolar electrode, as we have previously discussed. This comparison yields measurable electrical potential that form discernible EEG waveforms. So, now we know from where this EEG signals has been generated and how to capture them through unipolar and bipolar electrodes. Now, how we can record it? What are the, how to place these electrodes over the scalp? Is there any rule? Yes. So, prior to beginning to interpret the EEG, one should first ensure that the study was appropriately calibrated. Means before recording this, we must have to calibrate the electrodes by providing some known signal through it. Then this first passes a known voltage through the system to ensure that each amplifier for each channel handles this known signal identically. So, then comes to the electrode placement, where we have to place these electrodes. A very known or interesting, or we can say well known recording system or electrode placing system has been adopted, that is the standard international is 10-20 electrode system. So, 10-20 electrode system is like this. You can have, if this is your nasion and this is inion, so from nasion to inion, the whole place or whole distance is first divided into this. First we take the first part or first if it is your frontal lobe, F means frontal, then this is your central, then it is your parietal, then O is your occipital. So, now you mark from nasion to frontal, the first electrode which you can place here, it is at a distance of 10%. Then after 20% distance we are placing the frontal electrode, then from frontal to central it is again 20%, again from central to parietal 20%, from parietal to occipital again 20%, and from occipital to the inion it is your 10%. So, in this manner the whole 100% of this space has been divided. It is known as 10-20 electrode system. Now, in this case also you mark the anterior posterior dimension. This is from anterior to posterior dimension, that is in between the nasion and inion, that is a prominent bump on the back of head representing lowest point of the skull, and between the auricular positions. So this part is your auricular position near the ear. Each electrode site is represented with a letter and a given number. That letter is defining its positioning on that particular lobe, as we have discussed in the first slide. So it is your front, F is for frontal lobe, T is for temporal, P is for parietal, and O is for occipital, and in some cases you will find C is your central. So now odd number of electrode sites are on the left. If you divide this, if this manner, here you have divide this, so this side will find left side, and if it is your right side, on the left side you will find all the odd number of electrodes. You will find F3, then T3, O1, like that, and in the right side we will have even number of electrodes like F6, F8, F4, and Z is label site at the midline. If this is the system or the electrodes which we are placing in the midline, so you can mark here it is the central point or it is CZ. Central lip, this if this is your frontal lobe, and the electrode which is placed in the midline, it is your FZ. It is your parietal lobe, PZ, like that, anterior to posterior, and A is auricular site, auricular, or it will be placed at the near this ear. So it is your, these electrodes are named as so. Now different electrodes, we are considering those as I have discussed. We can take the electrode system, or we can take this, let's say from F3 we are taking one signal, and from T6 we are taking a signal. These two electrodes are recording two different signals, and their difference is now amplified through the amplifier or pre-amplifier. So in this manner we can gather, or we can collect many electrical signals, and after adding them, or after the combination of collecting these signals, then we can have this, basically these 5, and last one is your this 6. Here we have this 5 different types of signals or waveforms, electroencephalogram waves. So a typical adult human, these waveforms is about your, it is in the order of 10 microvolt to 100 microvolt, okay, in the amplitude when measured from the scalp, and they falls in the range between 1 to 100 hertz, not more than that. So first of all we will see some waveform, that is this one. It is your delta, it is your delta waveform. So this delta waveform, it is up from DC, means almost 0 to 4 hertz this frequency, and they have the highest amplitude. You mark here, this amplitude is high, maximum amplitude are getting for this delta waveforms. Then theta waveform, theta wave will having frequency 4 hertz to 7 hertz, and these are dominated when somebody or that person is at in some meditation, he is at drowsiness or some arousal. At this time we have the brain is if we collect those signals, they are generating theta waves. Now next one is alpha wave. Alpha, it is seen posterior region of the head on both sides. They have a frequency from 7 to 14 hertz, higher in amplitude. You mark here, these are your theta waves, this is your alpha waves. This alpha wave, they have higher amplitude, dominant side with relaxation, okay. At relaxed state of our mind, basically the brain is generating this alpha waves. Now next is your beta waves. Now this beta waves having higher frequency, that is your 15 to 30 hertz. It is associated with active, busy and anxious thinking and active concentration. Means if you are focusing, your brain is active, then this beta waves has been generated. The next one is gamma. Gamma wave is from 30 to 100 hertz. You mark the frequency is so high, and it carrying out a certain motor function. When some motor activity is going on, nervous system is active, then this gamma waves has been generated and captured. Now this is the comparison table of different EEG waves and when it is generated. So you mark if it is a delta band, as we have discussed, the frequency starts from almost DC, means 0.5 hertz to 4 hertz, and it generated at frontal lobes of the adults and posterior lobes, posterior position of the kids. They are normally deep non-rapid eye movement, NREM sleep, and high amplitude, and they have pathologically they have attention task, subcortical, diffuse lesions and metabolic encephalopathy. These are the pathological significance, or we can get some indication from this delta waves. Then we have theta. Theta wave has frequency from 4 to 7 hertz, and they are non-task regions, means somebody is not active, so they are actually in infant and drowsiness normally, and pathologically they signify the subcortical lesions, encephalopathy and deep disorders. So these are some indications, pathological indication we need to detect from this theta wave. Similarly alpha waves is frequency having 8 hertz to 12 hertz. They are generated normally in the posterior or head and dominant sides, and these alpha waves are generated at relaxed state and when eyes are closed, and they reduced and abnormal in neurological disorders. If they are reduced or abnormal, then we can detect that there must be some disorder in the neurological system of that person. The next one is beta waves. Beta waves are from 13 or 15 to 30 hertz. So they are generated with frontal and symmetrical locations, frontal lobe and symmetrical locations, and they are active thinking, alertness. These are the indication. If somebody is alert, is thinking something, then beta waves are dominated. And now if pathologically, if somebody have some anxiety, medication effect, then also you are getting some beta waves. Then gamma waves is more than this 30 or 32. It is location is somatosensory cortex. Then they are normally for perception and memory, but if this gamma wave, it indicates or gives a significant indication in case of your epilepsy and cognitive decline. And finally this mu is from, let's say, 8 to 12. It is sensory motor cortex. It rest state motor neurons, abnormal suppression in autism. This is the indication. So these are the some about the comparison of different EEG waves, their frequency, from where they are generated, and how they are indicating to different pathological conditions. Now how to capture this? We have already discussed different types of electrode placement in the different lobes of our brain. Now this is your block diagram of a EEG machine. Now you mark here the scalp electrodes, they are placed here and they are connected to a jack box. Out of this jack box, multiple number of electrode pins are connected to a electrode montage selector. What is that? Electrode montage selector. EEG signals are transmitted from the electrode to the head box, and which is labeled according to 10-20 system, and then to the montage selector. The montage selector on an analog EEG machine is a large panel containing switches actually. They allow the user to select which electrode pair will have signals subtracted from each other, and that selection or that selected electrodes, the signals which has been captured by them only are amplified through the pre-amplification, and it create an array of channels of output called this montage. So before selecting these electrodes, we need to calibrate through this electrode test by sending, we need to calibrate the amplifiers by sending the test electrode signals, and then once this montage selector is selecting and sending these sending those signals from two different electrodes to this pre-amplifier, then this pre-amplifier will amplify that. So every channel has an individual multi-stage SC coupled and very sensitive amplifier. Every channel has their own pre-amplifier with differential input, because from two different electrodes we are collecting the signal and we want to amplify that, and adjustable gain in a wide range. Its frequency response can be selected by single stage passive filter. So we will have different types of filters here. This after this amplification process, then we have this overall sensitivity of an EEG machine is the gain of the amplifier multiplied by the sensitivity of the writer. Once this signal which has been captured and amplified or pre-amplified, then they may contain some artifacts. As we know, artifacts means noises in the signal. Those noises has to be filtered out. We have different types of filter connected after this amplifier. So they have high pass filter, low pass filter and notch filters. So once the signal has been filtered, this noises has been filtered from the signal, this processing is called signal conditioning. Then after that we send that signal to some analog to digital converter and to the computer to process that signal and to monitor that. Otherwise we can also send the signal to a writer unit, and this writer unit we draw the graph on a chart drive through some ink writing oscillograph, and these are the different parts as we have discussed: filter, writing parts and paper drives. Then the channels, what we have discussed previously, an EEG is recorded simultaneously from an array of many electrodes. Those electrodes may be bipolar or monopolar. As we have discussed, bipolar means two different electrodes we are taking the input simultaneously, we are collecting the signal simultaneously. Otherwise in monopolar leads we can take the single electrode along with with respect to one reference electrode. Next one is this, basically the commercially available electrode system are 32 EEG channels. They have 32 EEG channels. They are available in the market. Now finally, through this electrode system, through this electroencephalogram, what we can capture, they have a significant importance in brain computer interfacing. So brain computer interfacing or BCI, using this EEG that enable direct communication between the brain and the external device by capturing and interpreting brain electrical activity in non-invasive manner. So these electric signals which has been captured through these electrodes or EEG, this based BCI extract features from neural oscillation related to intention or cognitive state, like as we have discussed in case of your beta waves, which are then translated into control commands. Those beta waves will be captured and translated or mapped to a control commands for computers, for prosthetics, for wheelchairs or for communication aids. So these systems have significant clinical impact for patients with severe motor impairment like spinal cord injury and locked in syndrome. But through their thinking, they can generate the signal and they can have produce some activity. Then other cases like epilepsy, seizure disorders, they can be also detected through the abnormal pattern of these EEG waves. Similarly, sleep disorder including narcolepsy and insomnia, by monitoring brain activity change during the sleep stages, they can be cured. It can be monitored and then can be treated. Similarly, brain tumors, head injury, strokes and brain infection, they also affect the normal active brain electrical activity. So, according to those activity, the waveforms will also degenerate. Neurological diseases like encephalopathy, Alzheimer's disease and dementia for functional brain assessment can also be detected and can be the treatment can be provided after this detection through this EEG waves. It is all about your EEG. So, thank you.